You can always tell it's Friday because nobody wants to get started. Right? I've got CMC coming to try to deal with this noise, which I find very annoying. I'm not sure what it is, but hopefully they do. It sounds like a mosquito somewhere in the wiring. I'm not sure. Okay, so we've pretty much made our way through protein structure. And what I want to do is try to answer the question I posed at the end of last lecture. The question I posed was as follows. I can take ribonuclease, which is very stable, and if I don't do anything else to ribonuclease, I boil it, it comes back together, and it works just fine. If I take ribonuclease and I treat it with mercaptoethanol, which converts disulfide bonds into sulfhydryl bonds, and I also treat it with urea to disrupt hydrogen bonds, and I boil it, what I discover that I can pretty much denature the enzyme. If I take all of those components out of there, I take the mercaptoethanol away and I take away the urea, what I discover is that a little bit or some of that enzyme will come back together and will still be active, which tells us, as I noted, that the information for folding properly is contained in the sequence of amino acids. There's nothing else external there that's causing that to happen, because the only thing in the tube is what I started with, the ribonuclease. Okay? Now, the question I posed was, if I take those components away but I leave a little bit of mercaptoethanol in there, what I discover is I can make more of the enzyme be active. In fact, I can make almost all of the enzyme become active again. And so my question was, how and why? Okay? And somebody came up to me after class and they actually had the right answer, so I'll throw it out to you and see how many others think you know the answer to that question. 1, 2, 3, 4. Does any of you want to be brave and say it out in front of your classmates here? Nobody? Yeah.
A little bit of mercaptoethanol to break down some incorrect cysteine bonds.
Yes.
And when it would reconfigure again, it would make go into a lower conformation or energy, and maybe it would drive it.
Right. So what he said was that a little bit of mercaptoethanol can help fix the incorrect disulfides that come back together. Now, there's a couple ways that this thing can come back and make the wrong disulfide bonds. One is just by bumping into each other. They bump into each other. Okay, this polypeptide chain over here has a sulfhydryl. This polypeptide chain over here has a sulfhydryl. They bump into each other. They're going to make a disulfide just fine, but that's going to keep it from folding properly. The other thing that can happen is during the folding process, there are many possible states. And in the normal process, when that enzyme is being made, it's being made one amino acid at a time. So there's an order to how it's coming together, and therefore there is some order to how those disulfide bonds can form. However, when you have a long chain that's already been made, and you say come back together, there are possible ways it can fold that would never have been possible in the original synthesis of the enzyme. Yes, ma'am? Okay, so she says, how do you control that folding when you have a little bit of that mercaptoethanol left? What you're doing is you're not controlling it. You're saying that when this thing folds and it gets to this disulfide bond, okay, this disulfide bond is broken by mercaptoethanol, so it has a chance to refold. Okay? It can. That's correct. Okay. So the folding process, as you already saw, will to some extent give you somewhat normal and active enzyme. The folding process is dynamic. That is, it's going on this whole time it's sitting there in the solution. So it can get to a state that was okay, or that allowed disulfide bonds to form that wasn't quite right. When you break that disulfide bonds, it's dynamic. So it still is trying to fold, still trying to do its thing, and it eventually finds the correct state. That's basically what's happening with that. So folding is an absolutely phenomenal and magical process. Yes, Lori? Why doesn't it break down the right disulfide bonds? The answer is it can and does. But the right disulfide bonds are only structural components. You can break the right disulfide bonds and the enzyme will still be active. Okay? So you can take a properly folded enzyme, treat it with mercaptoethanol, and the enzyme still breaks down RNA. But it won't be stable at high temperature. Remember, these are beams that are structural beams that are holding it together. So as long as I'm not putting it under any stress, the normal structure will hold. Okay? Good question. Okay, yes? No, the RNA is what it acts on. So the RNA has nothing to do with the enzyme. See, the RNA is the substrate that the enzyme acts on and breaks down. So it has nothing to do with the RNA. Okay, well, before I finish this topic, I want to tell you one other consideration. That other consideration is how immense the folding problem is. Now, I said we're pretty good at recognizing amino acid sequences that will give rise to alpha helices. And we're pretty good at recognizing amino acid sequences that give rise to beta strands. And we're also pretty good at recognizing where bends will occur. But I said we're terrible at being able to predict the 3D structure of a protein. You think, well, what the heck? If I know secondary, and I know there's bends, why can't I just put it in the proper position? I want to give you an illustration of a problem that I am not making up. Okay? An illustration of a problem that I am not making up. The problem is this. If you take, and the calculation has been done, if you take a polypeptide chain that has 50 amino acids in it, 50 is pretty small. 50 amino acid polypeptide chain, what's the word? Oh, yeah, I can't do that, so we'll just have to be annoyed. Okay. All right, thank you. The mosquitoes aren't going away. All right, so 50 polypeptide chain, a very small polypeptide chain. When you look at all the possible rotational angles and all the different ways that that a 50 polypeptide amino acid chain could bend, you can determine how many theoretical possible structures could exist. And so one notion about how folding occurs is to say, well, folding is a random process. We thought we had the evidence from ribonuclease, okay? Here it goes the wrong way, maybe it comes back and it folds the right way. Maybe there's a randomness to it and eventually it finds its proper thing, and that's fine. That's been pretty much disproven, and the reason it's disproven is the following. People have taken those numbers that they have calculated for all those different possible positions for a very, very small polypeptide chain. And if they take those numbers and they assume that one of those possibilities could occur every 1/1000th of a second, okay? Every 1/100000th of a second. And we, instead of doing it in a polypeptide chain, we had a computer doing it. We had the computer say, every 1/100000th of a second, look at this possible configuration, and then this possible configuration, and this possible configuration. So that in 1 second, you can do 1 billion calculations or 1 billion possible structures that would be there. If you took the most powerful computer on Earth, in fact, if you took all of the most powerful computers on Earth and you applied them simultaneously to the problem, okay, it would take 1,000,000 times the age of the universe to solve it. 1,000,000 times the age of the universe to solve the problem. Now, that's a pretty hard number to get your head around. It tells us that folding is not a random process. Okay? Folding is not a random process. We know that folding is driven by amino acid sequence, and we know some of the generalities of amino acid sequence, but getting that right structure, okay, is something that, as I said, we may not come up with computer answers for that. Right? It's one of the big problems of biology. It's called the protein folding problem because people would love to be able to determine protein structure simply from sequence. Can't do it. So as it is, instead, we have to look at using techniques like X-ray crystallography or nuclear magnetic resonance to determine structures of proteins. And these are powerful tools, but they're very time-consuming. Okay, not quite as time-consuming as 1 billion times the age of the universe for all the most powerful computers on the face of the Earth. Until upgrade 2.0 or something like that, I suppose. Okay. All right. The last thing I want to say about protein folding is something probably you've heard something about, and I want to mention it as well, and that is a very interesting phenomenon associated with problems with protein folding. The problem was recognized years ago in the form of some odd animal diseases. One was called mad cow disease, another was called scrapie that occurs in sheep, and it's now known that these type of diseases occur almost all the way through the animal kingdom as well as in human beings. The disease that occurs in human beings is called Creutzfeld-Jacob syndrome, and it is a debilitating neurodegenerative disease that results in dementia. It occurs pretty rapidly, and the person dies of that. Cows, sheep, all the other organisms that get this all have the same related type symptoms. They're all having problems with nerves that are basically being destroyed, and they go crazy in the process. It was shown back in about the 1980s that this thing that caused this disease was very unusual. No matter how hard people looked to find the virus or the bacterium that caused it, they could find nothing. They could find absolutely nothing. We can find viruses anywhere, we can find bacteria anywhere, but nobody could pull up anything out of this. And it was very puzzling. And a man named Stanley Prusiner, who everybody laughed at when he proposed it, and who later won the Nobel Prize for his proposition, said that the problem is that this is not an infectious thing like a virus. It's not an infectious thing like a bacterium, but instead the infectious thing is a protein. And that was, as I said, ridiculed. It was greeted with scorn because the notion that a protein could be infectious all by itself really seems to violate everything we know about biology, because with viruses and with bacteria, they all have DNA, RNA genomes that allow them to reproduce. A protein all by itself doesn't have information to reproduce itself. How could a protein be infectious? But that's what all the data said, and he said that's what it says. It turned out he was right. And it turns out that the protein is not, in fact, an infectious protein like a virus that goes from one thing to another. It is infectious, and it is a protein, but it's your own protein. Okay? It's your own protein. So there's a protein that we all have in our brains that is in the brains of every animal. It's even been found in things related to it in organisms like yeast that are unicellular. Okay? This protein, in its normal state, has functions in the brain that allow the brain to function that aren't completely understood. But what's known about this is that this normal protein, you say, okay, now I understand. I get a mutation in this protein, in the sequence that makes this protein, and now I make a bad protein. And the answer is that that's not the case. It's known that mad cows, for example, don't suffer from a mutant protein. They have a 100% normal protein, at least in terms of amino acid sequence. What happens is that they have this normal version of the protein that has made an error in its folding during the synthesis. And when it makes its error in folding, needless to say, there's devastating consequences, but the way in which this thing happens is really remarkable. It appears that the misfolding of this protein induces identical copies of it to misfold as well. Okay? So that's why it appears like it's an infectious thing, because what it's doing is that once it gets started, it doesn't stop. It keeps multiplying itself. More copies of the protein get made, more misfolded copies of the protein get made, and what happens is you develop in your brain what are called plaques, which are these giant collections of these proteins that are misfolded, as you see here. Here's the normal one, here's the misfolded one shown here. These plaques actually destroy nerve cells, and that's what gives rise to the dementia that people have, that cows have, etc. Now, the question arises is how infectious are these from one person to another? How infectious are these from one organism to another? And it's a topic that's somewhat controversial. But there's evidence that there may be, there's some evidence that you may be able to contract, for example, mad cow by eating beef. Okay? Now, in this country, supposedly, we inspect and monitor all of our beef supply. This doesn't happen, although the inspection rate is pretty low. The reason the evidence that this comes up is that in Great Britain in the 1980s, okay, they had a mad cow outbreak, and they did not, in fact, have an inspection system, so that meat from mad cows made it into the British food supply. And a few years later, they had a great increase in a form of Creutzfeld-Jacob's syndrome that still resulted in death, but it wasn't exactly like the original. And the thought was perhaps these people, and because of the large number of increase in people who got this, perhaps what had happened was this had gotten in the food supply, and they had, in fact, contracted from there. It's controversial. As I said, there's not establishment for it, but the numbers are a little disturbing. The perhaps even more disturbing fact about this is the stability of this misfolded protein. You might say, well, I cook my food. I don't have to worry about it. And the answer is, you do. Okay? Because what it takes to destroy this protein or unfold this protein by heat is typical temperatures of about 700 degrees Fahrenheit. Most people don't cook their food like that. And if they do, they probably have more danger from the DNA adducts that they created in burning it than anything else. Okay? So that fact alone has driven many people to vegetarianism. I'm not advocating that. I'm just saying that that's what has happened with many people. Yes?
I was just going to ask about, so the information for cooling has to do with amino acid sequence.
That's correct.
Why is it
Okay. It is related. Her question is, well, if this thing is misfolding, why isn't it folding like the proper protein? The answer is that there are multiple ways of folding. We sort of saw this with ribonuclease a little bit, where we could misfold if we didn't put things in the proper context, and probably that's what happens here. Okay? So it's probably in some way getting a misfold, and once it starts misfolding, you're hosed. So there are many different states of folding, and so this is probably at the level at which this is operating. Okay? And your body has misfolded proteins and probably normally just discards them, or because they're not functional, they just sit around and do nothing. This one we know about because it multiplies. That's how we know about it. Yes, Lainey? They're all misfolded in exactly the same way. Yep. Yep. Yes?
So are these already folded proteins that are then refolded into the stack shape, or is it just new ones there?
Well, it's kind of a hard question to argue, because once you get one, it keeps making more that do that. So did it start out okay and then flip, or did it misfold on the way to being okay? It's not a question that's easily answerable.
I don't think the original one,
Yeah.
the ones that it makes.
So it makes them fold improperly as they're going through. It doesn't get here and then go do that. As it's folding, it interrupts the process and makes it fold like that. So it's an error during the folding process, not something that it takes apart and refolds, but an error that occurs during folding. Okay? Yes, question back there. Yes? Gene expression? Very good question, okay? Is this something that why is it that it's making more of the protein? Okay? I don't have a good answer for that, but what may well be happening is when the cell doesn't have enough of a protein, it starts making more to compensate for it. And so it's making this protein for a reason. It needs this protein. And so when it doesn't have proper protein, what is it going to do? It starts cranking out more. Yes? I don't have an answer for that question. Clearly, this guy is pretty resistant to enzymatic attack. So it's an extraordinarily stable protein. I don't have an answer for why it's not attacked by proteases. Yes, Laurie. Okay. I don't mean to say it folds differently every time. It's not a random thing, as I said. But I'm saying there's possible ways in which it can do other things. That's clearly what's happening here. And for the most part, you're never aware of it because you misfold a protein that's in your thumb and it doesn't function. The cell just eventually deals with it and takes care of it. You don't know about it. You know about this one because A, it duplicates itself, and B, it causes a problem. So you don't know that about a regular misfolded protein. Now, this is leading me to one more question here, and then I'll move. Yeah. Yeah. Did I what? No, it's not known what may instigate the misfolding. It's not known. Okay, so we don't know what may cause the initial misfolding to occur. What we do know is once you've got a misfolded one, you're going to misfold more. That much we do know. So the concern is if you're getting it in diet, could that be a problem? You know, that's a thought. Okay. Now, folding of proteins is clearly very important. And so because of the concerns, now I put this idea in your head that protein folding is random and so forth, which I've tried to convince you it's not. It's not random. Okay? But you can see that there could be some problems with proteins that misfold. And so cells are ahead of us in that scheme, and that cells have structures of proteins called chaperonins that actually help proteins to fold properly. I know you're going to say, why don't they work here? And the answer is I don't have that answer. Okay? But chaperonins are little complexes that allow a protein to fold properly. And how in the world does a complex allow a protein to fold properly? Let me give you an example that might make sense to you. Let's imagine that I am a ribosome. A ribosome is the thing that makes the proteins. And I'm scooting along the messenger RNA, reading the sequence of nucleotides, and I start putting together amino acids, and this amino acid tail starts coming out of me. Okay? And that's what happens during the synthesis of a protein. A little amino acid tail starts coming out of the ribosome. That amino acid tail has, okay, amino acids that have side chains that have chemical properties, right? And what have I told you about hydrophobic amino acids? Well, they don't like water, right? And where are they being squirreled out into, out of the ribosome? Right into water, okay? So what do you suppose they want to do? They want to avoid water. How can they avoid water? Well, one way they can avoid water is by folding. But the problem is the whole protein isn't there, so it's not able to completely cover those up. Those hydrophobic residues are still sitting out there, so that if behind me on the same messenger RNA there's another ribosome that is scooting along making exactly the same protein, it's going to have exactly the same hydrophobic residues sticking out there. Do you suppose they may associate with each other? You betcha. And if they associate with each other, they will not fold properly. This is probably the reason why cells have chaperones. So as to keep things that would otherwise not fold properly isolated from each other. That's what the chaperones basically do. They provide a little chamber into which the protein can be put so that it's not interacting with other things that cause it to misfold, and it folds on its own. Very cool thing. So chaperones are involved in that, and I'll spell that for you. It's C-H-A-P-E-R-O-N-I-N. Chaperones. Okay. I think that's what I want to say here. Yes. Oh, I know. I was going to show you this one other thing. Some of you had asked about amino acid sequence and secondary structure. This table shows a ranking in terms of the likelihood of different amino acids to be found in alpha helices, beta strands, reverse turns. Higher the number, more likely. But that by itself can't be taken totally by itself, okay, because of the fact that it takes a sequence of 3 of these in a row, okay, to indicate likelihood. So if you, one of the rules is if you have 3 amino acids in a row that have a ranking over 1.0, the likelihood it will appear in that structure is high. So for example, if I had glutamic acid, alanine, leucine in a row, the likelihood it would be part of an alpha helix is pretty high. I can do similar things with beta strands. I can do similar things with reverse turns. And as I said, it's not exact, but it gives us a pretty good idea about where things lie. Yes?
Is that first term the same as helix, turn, helix, or is this?
No, it's the turn I showed in there. So a turn could be part of a helix, turn, helix. But a helix, turn, helix includes a helix, a turn, and a helix. Okay? So the reverse turn is just the bend itself. Okay? Good question. Okay, so that's what I want to say, the last I want to say about protein structure. Unless there's other questions? Okay. Well, let's turn our attention to protein characterization, for which we're going to now use some of our structural knowledge to understand proteins at another level. Most of what I'll be talking about here involves purification of proteins. And I'll also be talking about some ways in which we study protein structure. That will occur on Monday. Okay? But protein purification is something that, if you're a biochemist, you can't be a biochemist and not be interested in protein purification, because proteins, as I always like to say, are the workhorses of the cell. And whenever we try to solve a biological problem, it ultimately comes down, in almost every case, to the protein. Biochemists believe in the principle of divide and conquer. And that's not in the principle of how they teach their students, because I don't think that's a good way to teach, but rather in dividing the cell and purifying components so that you have pure components to work with, and then you can understand the function if you only have a pure component to work with. Understanding that function of one thing is a lot easier to do than if you have 1,000 things that are mixed together and you think that whatever you're seeing happening is due to the one thing that you're interested in, but it may not be. So we have to purify. We have to get proteins isolated from each other. And being able to purify proteins is a very important and useful tool. Okay? Your book, for bizarre reasons, talks about lactate dehydrogenase, which has nothing to do with protein purification. I'm not even going to bother with there. Instead, when you go into any biochemistry laboratory, one of the first things that you'll always notice, it's almost impossible to find a laboratory that doesn't have a centrifuge. So centrifuges are critical for us to being able to isolate components from cells. If I'm interested in a protein, let's say I'm interested in hemoglobin from blood cells, I could take a bunch of blood cells, I could go grind them all up, or I could treat them with a sonicator to bust them open or whatever, and then all of that mix of that cell would be contained in the tube in which I had just busted open these cells. Centrifugation is almost always the first step in extracting or pulling out the things that are of interest. So centrifugation allows us to use centrifugal force as a sort of artificial gravity to precipitate things. You guys probably played with these in chemistry labs and so forth. The faster the rate with which you spin a tube, the smaller the things that you can precipitate with that. And things that don't precipitate at a given centrifugal force end up being what we call the supernatant. Okay? All right. So I can take and spin at low speed, and I can spin down, for example, cells in the first place. I can take those cells, I can bust them open, I can spin them down at a higher rate, and I can isolate cellular organelles like nuclei, like mitochondria, or like pieces of cells. And that's what we see in all of these cases right here. Okay? If the thing I'm interested in is in the nuclei, there's what I'm going to go work with here. I'll take the nuclei, I'll bust them open, and I'll get the protein out of there. If what I'm interested in is the mitochondria, I'm going to take it out of here by doing the same thing. It may be components of cells, like membranes or structures inside of cells that the protein is embedded in. Or last, it may simply be dissolved in the cytoplasm, in which case it's going to be in the supernatant after I've done all of these processes. Okay? So I have to go looking through. And in some cases, when I start purifying the protein, I don't know where it's going to be. I don't know. Is it here? Is it here? Is it here? Is it here? And since I'm trying to purify it, I don't even exactly know what it is. All I know is what it does. Okay? So in the case of hemoglobin, if I'm trying to follow hemoglobin, one of the things I know hemoglobin does is it binds oxygen. So I could put an oxygen sensor into a tube. I could take a little bit of this extract, 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 4 different tubes, and see which one of them, when I put oxygen in it, gobbles up oxygen. What I've just described to you is a way of determining where my protein is, what extract my protein is contained in. So a critical component of protein purification is having what's called an assay. An assay is simply a measurement of the protein's activity. In this case, binding of oxygen. Okay. Now, the other techniques that I use depend upon which of those extracts I pulled out of. But in each case, I'm going to have an extract that's going to have my protein almost always dissolved in water after I've done something to it, whether I've busted open the nuclei or busted open the mitochondria or something like that. Okay? Well, busting it open, of course, doesn't guarantee purification because there are a lot of proteins that are found in the nuclei, thousands of them. There are thousands of proteins found in the mitochondria. Okay? Thousands of proteins found in the cytoplasm, thousands of proteins found in any of these different fractions that we just saw. So the other techniques are used on top of, in addition to, the centrifugation. So one technique that's very useful, you probably played with it a little bit in biology lab, is dialysis. And dialysis is simply a method of removing small molecules from big molecules. The small molecules here are very small molecules. Typically, they're salts. So on the left, we can see an example of a mixture that I had that had a lot of small molecules in it. It also had my protein in it, which is shown in red. If I take and I put this into a membranous tubing called dialysis tubing and close off the end so that it can't leak in, when I put that in a mixture that has a buffer in it, what's going to happen is the small molecules will come out because they're smaller than the openings in the tube. They will diffuse so that the concentration of the small molecules will basically equal each other around the sides. The large molecules, the proteins, are too big to get through those holes, so they stay inside. And I've therefore removed an awful lot of the small molecules from my protein. So dialysis is very straightforward, it's very simple, and it works basically on osmotic pressure. A more powerful and interesting technique is that of gel filtration. And for gel filtration, we rely on some interesting chemically modified beads. Okay? So these little beads are little tiny things about 1 millimeter in size, maybe smaller. Okay? They're literally little spheres. And these little spheres have been made in a very interesting way, and no, I can't tell you how they're made, but I can tell you that they are made. They're made in such a way that they have little tunnels going through them. Little tunnels. And those little tunnels have holes that open and close. You open one end, go out the other end. Those little holes have all the same exact size, and the tunnel itself has the same size as the hole that's the opening for it. Okay? Everybody envision that? So you got a little bead. It's got a little tunnel running through it. That little tunnel has a diameter, as it were, that has consistency throughout it. Now, if I take a column like this and I pour into there these gel filtration beads, and I start adding buffer to let buffer flow through those beads, of course, buffer will fill up the column, it'll fill up the beads, and it'll flow all the way through there. Then if I apply my protein sample to the top of that, something interesting happens. And the something interesting that happens depends on the size of that little hole that was in the bead. Let's imagine I've got 3 proteins. I've got baby bear, mama bear, and papa bear. Okay? Baby bear is pretty small. Mama bear, not quite so large. Papa bear's been hitting Thirsty Thursday. Okay? He's got a bear gut, I guess we could say. Huh? I just made that up. You got a bear gut. Okay. I'll have to remember that. It's Friday, right? Okay. So, mama bear, papa bear, baby bear. All right? Papa bear is way too big to get into any of those holes. What's going to happen with papa bear? He's going to hit a bead. He's going to bounce off. He's not going to enter any of the beads. Mama bear has been working on her figure, and maybe she hasn't quite got it the way she would like, but she's tight enough, she's small enough that she can sort of squeeze in there if she kind of sucks in her gut and make it into some of those holes. Baby bear sees this like a little carnival. I want to ride every ride. Goes into every hole, travels every single thing. The distances that the 3 of these bears are going to travel and going through this column are very different. Papa bear is just going to bounce, bead to bead to bead to bead to bead out. Papa bear is going to be the first one through. Mama bear is squeezing through. She's going to take a little bit longer route than Papa bear. She's not going to make it through quite as fast as he is, but she'll come out second. Baby bear, who's at the county fair and seeing all the rides and doing all this stuff, is going through every single one because baby bear has no trouble fitting into any of those holes. As a consequence, comes out last. Gel filtration is a way of separating proteins on the basis of size. The largest proteins come out first. The smallest proteins come out last. That hole that I described to you in those beads is known as an exclusion limit. The exclusion limit is related to the size of the protein. The exclusion limit is usually given in terms of molecular weight. So an exclusion size of, let's say, 25,000 will exclude, on average, proteins that are larger in 25,000 molecular weight, but let those of under 25,000 molecular weight enter and do the baby bear thing. Everybody with me? Okay, so it's a cool technique, cool technology. Question on that? Okay. Next step in the process, there are other beads that we can have. Okay, so let's say I've got a bead, but instead of having little holes in it, I chemically modify the surface. I chemically modify the surface so that I put an arm that sticks out on there that's got, let's say, carboxyl groups. All right? And so an individual bead might have thousands of these arms, each of which has carboxyl groups on it. Okay? When I ionize a carboxyl group, what charge does it have? Negative. Okay, good, good. I was hoping I'd hear some answers on that. Right? I've got a bead that's full of it's got a coating of negative charges. Okay? If I have a column that's full, and this is an example of a column that has negatively charged beads. If my column is full of negatively charged beads, what do you suppose is going to happen when I put in proteins, some of which are positive, some of which are negative in that column? Which ones are going to come through first? The negatives, of course, because the negatives are not going to stick to the negatively charged beads. They're going to come shooting through. The positives are going to be attracted, and maybe they'll wait there for a little bit, and then they'll slide over here, and then they'll slide down here, etc. But they take a lot longer to get through the column. Okay? What I've just described to you is what's described as a cation exchange column. It may seem a little backwards because I'm talking about negatively charged things, but it's cation exchange because remember I had to ionize to begin with. It started out with a neutral charge, and typically it has sodium instead of hydrogen. It's not starting out as an acid. It's starting out as a sodium on there. The sodium comes floating off. That's what leaves behind the negative charge that allows the protein to stick on there. The protein is replacing that sodium, so it's cation exchange. A cation exchange column will bind to positively charged ions. It will push negatively charged ions through. Okay? So the positives will come through last, the negatives will come through first on a cation exchange column. Similarly, I could create a column that has arms, and instead of having carboxyl groups, it could have amine groups, in which case it's going to be positively charged. And it's going to have counter ions that typically are chlorine, which is a negative ion. Columns such as this, when the chlorine washes off in solution, leave positive behind. It will stick to negatively charged things. This is known as an anion exchange column. An anion exchange column is exchanging that negatively charged chlorine for a positively charged, or for a negatively charged protein. Okay. Now, so cation and anion exchange proteins are very, very useful for purification purposes because they allow us to separate on the basis of charge. Okay. Questions about that? Yes, Lori. Okay, so will it separate something on the basis of zero charge? The answer is to some extent it will, because you've got things that come through very fast because they're repelled, you've got things that come through very slow because they're attracted, and somewhere in the middle will be the less charged molecules. So yes, to some extent that will do that. Okay. All right. There's what some of the structures look like. There's an amine, there's a carboxyl, blah, blah, blah. There's the bead. Okay. Now, one of the more interesting types of chromatography uses a bead that's been modified in a different way. This bead has been modified to contain a molecule. Okay? Well, how does this work? Well, let's say I'm a researcher in the laboratory, and I'm very interested in proteins that bind to GTP. Okay, GTP is one of the nucleotides. I am interested in identifying and purifying proteins that bind to GTP. So what do? Well, I take those little beads that instead of drilling a hole in them or instead of putting a charge on them, instead I attach to that arm of the bead, I attach GTP. So now I've got a bead that's got GTP sticking all around it. I take my mixture of proteins from my cell that might have thousands of proteins in it, and I pour them in there. What's going to happen? Well, what happens is that proteins that bind to GTP are going to latch right onto that thing and stay there. Proteins that don't bind GTP see nothing of interest here, and they just come shooting through the column. Now, this type of column is generally fairly specific for the thing that it binds. In the case of GTP, you'll actually find a bunch of proteins. But in some cases, you might be looking for a structure that only one protein binds to. There's a dandy way to purify your protein out of a cell. You make this is called an affinity column. You make an affinity column for that structure that you're interested in, and bang, you got that protein pretty easily. So affinity chromatography uses a molecule or some molecular structure that a protein binds to as its attachment to the bead support. noise really gets to you after a while, doesn't it? That makes sense? Clear as mud? You guys look tired today. Are you tired today? Are you ready for the weekend? I don't have a song for you today. I could tell you a joke. Okay, how about a joke? All right. Let's see, which joke should I tell you? I'll tell you a dumb joke, okay? Okay, this is my joke about Artie the Hitman. You ever hear about Artie the Hitman? Has anybody ever heard about Artie the Hitman? You wouldn't tell me if you did, would you? Okay. Artie the Hitman. This guy decides he wants a career to be a hitman. He wants to kill people for a living, right? Takes an ad out in the local paper saying, you know, aspiring hitman, you know, will work for cheap, okay? You know, and he decides to really get interest, he'll do it for $1, okay? So he puts this ad out there, and he gets a call. This guy calls up and says, yeah, he says, could you knock off my wife? Artie says, sure. He says, $1, right? He says, yeah, $1. He said, where can I find her? He says, well, she goes down to the grocery store a lot. I'd like you to knock her off down there. No problem. Okay. And oh, by the way, could you strangle her? Sure. You know, he figures to make a name for himself and so forth. So he gets the $1 and he goes following the wife and finds the wife down in the grocery store. Okay. And he looks around. There's nobody around. He walks up and he grabs her and he strangles her right there in the grocery store. And she falls dead. And he goes, yes. Uh-oh. And somebody saw him. Okay. So somebody's seen him and he goes, I can't, you know, this is going to stop my career right here. So he goes over, he grabs the person, okay, and he strangles them right there in the grocery store. Whew. It was close. Oh, not my day. There's somebody over here, okay? He goes over and he strangles them right there in the grocery store. He figures, no more, I'm getting out of here. So he takes out, goes running out. The police catch up with him. The headlines in the newspaper the next day reads, Artie chokes 3 for $1 at the grocery store. Ah, I had to work hard for that one. No, please. Don't encourage me, okay? Don't encourage me. I have more biochemistry to do. Okay, Artie chokes 3 for... I'll see later in the term, I'm just going to say Artie chokes 3 for $1 at the grocery store. I want to see if I get that same response, okay? Just hold that, okay? Just remember that. Okay, back to the exciting field of chromatography. We've talked about centrifugation. We've talked about dialysis. We've talked about gel filtration chromatography. We've talked about ion exchange chromatography. And now we've talked about affinity chromatography. It's time to turn our attention to 2 more types of chromatography. One is called high-performance liquid chromatography, or HPLC. People frequently, and I think your book even frequently misnames this as high-pressure liquid chromatography. The pressure people mistake it for is the fact that it uses a lot of pressure, but the correct name is high-performance liquid chromatography. This is a type of chromatography that's relatively new, meaning it's really become extremely popular in the past 20 or 30 years. And it also uses a sort of a bead, okay? These beads are much smaller than the beads I was talking about before, okay? And they're packed into columns extremely tightly, which is why it takes high pressure to push the liquid through these. So where you might have a few million beads in a regular column or a few thousand, you might have millions or billions of these little beads, these little tiny guys in a column of the same size, okay? Now these beads are different. They have arms that stick off of them that are not ions. They're not molecules that the substrate binds to, but they're typically long chains of carbons. Okay? Long chains of carbons. Those long chains of carbons are a fairly nonpolar tail. Okay? So this bead has this long nonpolar tail sticking off of it. When I apply a sample to this column, I'm going to see some things happen. Some of the things that will happen are that the nonpolar molecules are going to associate with these arms a lot. The polar molecules, like things that have ionized or that have great affinity for water, are not going to associate with that, and they're going to go right on through. What I've just described to you is a phenomenon known as reverse phase chromatography. It separates on the basis of polarity. Not on the basis of charge, but on the basis of polarity. The less polar things will, in fact, stay on the column the longest. The most polar things will come off of the column first. Yes? It's called reverse phase chromatography. Okay? All right. So, I'm sorry? What's that? So the least polar molecules come off last because they're associating with those nonpolar arms. The most polar molecules come off first. Now, there are other types of chromatography that are used for HPLC. Reverse phase is by far the most common, and that's what I'm going to hold you responsible for. I'm not going to talk about the other types of chromatography that we have in HPLC. Reverse phase. Okay. The last thing I want to talk about today is a very brief description, and I'll start with this next time, about gel electrophoresis. Okay? Gel electrophoresis is yet another type of chromatography, and it involves a gel. How many people in here have run gels? A lot of people have run gels. Okay. So gels get their name from the fact that it is jelly, gel-like. Okay? We typically make these from a support of either agarose, which is a seaweed byproduct, or polyacrylamide, which is a chemical that we actually make in the process of constructing the gel. Agarose is typically used to separate fragments of DNA. Polyacrylamide is typically used to separate fragments of or proteins, which is a fragments, but proteins. Okay? So I'll talk about polyacrylamide gels next time. I want to talk about agarose to finish out the period, and then we'll finish. Agarose simply provides a support, and a support means it's just a solid substance through which all the buffer and all the water is present. All right? Molecules move through this support not without additional help. In the case of HPLC, the additional help that we had pushing things through was high-pressure solvent being pushed through it. We don't use pressure here. Instead, we use electricity. So the driving force for gel electrophoresis, as its name implies, is electricity. If I think about a DNA molecule is a long rod, double helix. It's full of phosphates, so it's negatively charged. And if I put a series of DNA fragments at the top of a gel, some that might have 1,000 base pairs, some that have 500 base pairs, some that have 100 base pairs, and I put them at the top of that gel, and I apply electricity across that gel, putting the negative at the top and the positive electrode at the bottom, what's going to happen? These negatives are going to be repelled by the top, they're going to be pulled towards the bottom, and the speed with which they're going to go through is a function of their size. The smallest guys are going to make the fastest run through there. The largest guys are going to make the slowest. As a result of that, I can separate molecules on the basis of their size. Okay, I think that's a very good place to stop. Let's stop there, and I will see you guys on Monday. Have fun this weekend.